Hello everybody, welcome to the Integumentary System video lecture. So let's go ahead and get started. I'm going to skip the questions addressed here, but you can pause and read them if you'd like. So in the first part of our outline, we're going to go over the different layers of skin and also the functions of these various layers, as well as looking at how you have sensory receptors in certain layers of the skin and the different types of sensory receptors. So let's start off with just the basics here. So your skin is called integument, and your integument is composed of two major layers, the more superficial layer called the epidermis, and the layer just beneath the epidermis is your dermis. There are some layers underneath that you may see in the picture here, for example, the hypodermis, and then there's another layer of skeletal muscle, and under the skeletal muscle you'd have the skeleton or the bone structures, but the only two layers that count as your skin are the first two, epidermis and dermis. In terms of overall functions, there's at least four big ones that we can name. Protection as a main one, so there's certain kinds of tissues in your skin that help essentially provide some physical protection against abrasion. But there's also protection in the sense of your immune system cells that hang out in some parts of your epidermis, but mostly in your dermis. So if something like a thumbtack were to penetrate through the skin and that thumbtack had some bacteria and viruses on it, your immune system would be able to protect you to some degree from those pathogens. A second function would be temperature regulation. So, for example, sweat glands secreting sweat, which helps you cool down through the process of evaporative cooling. Third function would be vitamin D synthesis. So using UV rays, specific types of UV rays really, to help your body synthesize that necessary vitamin. And lastly, sensing different kinds of sensations, for example, temperature, pressure or touch, and also pain. In this slide, we're going to look at a sort of summary of the various types of receptors that you have in your integument. So let's start off on the left side here. So first we got Merkel cells. Merkel cells extend into your epidermis, but some of them are kind of hanging out in the border here between the epidermis and the dermis. And essentially these are modified neurons with specialized dendrites at the end that can detect light pressure. This one here looks really similar to the Merkel cells, but they're lacking these orange tips here. Those are just free nerve endings. So those are just kind of typical dendrites, you could say, and they're capable of detecting pain, heat, and cold. Let's see over here, there's some dendritic endings that wrap around your hair follicles. Those are called hair follicle receptors, and those detect touch. But they're going to detect touch a little bit indirectly. It's more like if a bug or something moves your hair on top of your skin, then the vibrations and the movement transfers through the hair follicle, and from the hair follicle that movement's going to translate into some action potentials through that neuron. So they're good at detecting touch. Over to the right here, and located at the top of the dermis, we have these Messner's or tactile corpuscles, and they're good at detecting light pressure. So if you can imagine some weight is pushing down on the skin here, it's going to squish the epidermis down, and eventually that pressure is going to hit these tactile corpuscles. In blue here, there's 4. And then they will send their action potentials to the rest of your nervous system. Lower, we have these receptor or these dendritic endings that look a little bit like a dicot, if you know what that is, a sort of like a two-leafed plant. These are called Ruffini endings, and they're best at detecting stretch. So if you can imagine this, if your skin was stretching left and right, some of the skin might be pulling on this little leaflet looking thing. It'll be pulling it to the right, whereas maybe the skin stretching over here is stretching to the left, and it'll pull this little dendritic leaflet over to the left. And when these two leaflets are separating from each other, that activates some action potentials. Then lastly, we have a Pacinian corpuscle, also known as a lamellar corpuscle. Those look a little bit like a slice of shallot or a slice of an onion. And those dendritic endings are specialized for detecting vibration. And they're typically also kind of in these lower ends of the dermis, and in some cases even in the hypodermis, like you see in this corner of the picture. Now, in reality, while I've pretty much said that they're detecting a couple of things, right? Hair follicle receptor is touch, Merkel cells are light pressure, etc. They're all capable of detecting all of them. For example, free nerve endings can detect some light pressure. However, they are very specialized for specific types of sensory stimuli. So I'm kind of simplifying things by saying they detect one thing, but hair follicle receptors could detect vibration too, not just touch. It's just specialized for touch. Now, something that's left out of a lot of pictures about your integument are the lymphatic vessels that flow throughout your dermis in your skin. So I had to pull this picture from a research paper. It's showing your epidermis in yellow, and then the second layer of your skin, deep to that, your dermis. Here are the green lymphatic vessels shown there. And the purpose of the lymphatic vessels is for essentially your immune system, and for your immune system to be able to detect pathogens more easily, and also so you can respond to some sort of foreign invasion more quickly. Now, they also are going to have an important function in terms of tattoo removal. So what I want you guys to do on your own time, because I can't quite record it with this video, but if you click on this link or type in this into YouTube, I would like you guys to watch the following time stamps from 0 minutes to 5 minutes and 30 seconds, and from 6.5 minutes to 7 minutes and 47 seconds. And I want you to think about, at the cellular level, how does a tattoo removal actually work? And then you'll be potentially tested on this concept. So, time to go into the more detailed view about the epidermis and the dermis, and talking about the sublayers in each of these categories. So we're going to go through them one by one. You can see the epidermis has 1, 2, 3, 4, up to 5 different layers in the epidermis, whereas the dermis has 2 major sublayers. In terms of remembering them, there are some acronyms out there. For example, come let's get sunburned, CLGSB. That will correspond to the different names, stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum basale. So that might help you remember the order, most superficial to most deep part of the epidermis. And in the dermis, I put this other picture here, a guy saying, people reapply, as in like reapply your sunscreen. The P stands for papillary dermis, and the R stands for reticular dermis. So all in all, we have up to 7 layers in your skin. And I kind of imagine these two maybe even having a little bit of an argument or not about whether to wear sunscreen or not. So let's start with the most superficial layer. The stratum corneum is also known as the horny layer, and this is composed of 20 to 30 rows of cells, though it might not really look much like it. The reason why it doesn't really look like cells is because they're mostly dead cells. The cells were called keratinocytes. Keratinocytes because they're cell types that are really good at making keratin fibers. And the reason why they're dead is they made so much keratin and they ended up being pushed to the more superficial layers, and now they're farther away from the blood. They're farther away from the capillaries of the dermis. Being too far from blood vessels means they don't have enough nutrients to survive, and especially when they're working so hard to make so much protein fiber, they need a lot of energy, so they end up dying, and their cytoplasm is just essentially replaced entirely with keratin fibers. They kind of explode in a sense. Overall, the function of this layer is going to be to have that keratin that's kind of out in the open now to prevent, protect against some friction and also help a little bit with the waterproofing of your skin. On the next layer down, this is one that's not present in all skin areas. It's only going to be present in the thick skin. So for example, the skin that you have in your palms where it's thicker, and also in the skin of your heels or your foot soles. So those areas that have more thick skin, they're going to have the stratum lucidum, also known as the clear layer. And this has about 2 to 3 rows of cells. Again, mostly dead cells, dead keratinocytes. And the function of this layer is just an additional layer of protection, especially because it's in your thick skin areas of your body. It's just extra 2 or 3 rows of keratin fibers to make it tougher and also a little bit more waterproof. Third layer deep, this is stratum granulosum, also known as the granular layer. Not an exact number of rows that we can say here, but a few rows of cells. And in this case, the function here is protection once again. Once again, it's going to produce those tough keratin fibers. But not just that, those cells are going to also produce lipids and release those lipids into the extracellular matrix. They'll release those lipids through exocytosis, and the purpose of the lipids is to help repel water. So when people say your skin is waterproof, the reason mostly it's waterproof is because you have some hydrophobic substances surrounding the cells of your skin. And they're there because of exocytosis. At this layer, the cells are close enough to the blood vessels that they are actually alive. So we'll have the living keratinocytes, in this case, actively forming intracellular keratin. So they haven't yet exploded. So fourth layer down here, we have the stratum spinosum, also known as the spiny layer, composed of several rows of cells. The cells are living keratinocytes. They're forming intracellular keratin here, but there's also different kinds of cells called dendritic cells that can be found at this layer. And they're like your early immune system alert cell. So they're one of the first cell types that will encounter a pathogen if, say, a thumbtack were to pierce to this layer. So here's our thumbtack that I kind of added to this picture. So here's the needle of the thumbtack. So say if there's bacteria or viruses there, if they were to hit through this layer, then your immune system, your adaptive immune system, might get stimulated after kind of capturing that pathogen. So in this layer, there's a protective function as well. Desmosomes, remember those specific cell-cell junctions that are really good at um, connecting cells somewhat tightly, but allowing them a little bit of wiggle room so that they can stretch a little bit and not rip apart. Also some immune system protection as discussed with the dendritic cells. The most basal layer is of course called the basal layer or stratum basale. Just one row of living cells here. The cells at this location are going to be really active because they're super close to the blood vessels, the capillaries, providing them many nutrients. So here's where we're going to find these stem cells that are constantly undergoing mitosis, which is cell division, and making new cells that will replace the old cells that slough off of your skin over time and make up, dust, dandruff, all that. Besides the stem cells being in this location, there are also melanocytes. And these melanocytes are what provide people with the different skin pigments. So some people have melanocytes that form more pigmentation and thus have darker skin, whereas other people have melanocytes that make less pigmentation and more white colored skin or paler skin. Specifically, the pigment is called melanin. And the purpose of the pigmentation has to do with shading the nuclei of your skin cells. By having the pigments surrounding the nucleus of those skin cells, it's essentially protecting the DNA of those stem cells, shading the DNA so that any UV radiation might not harm that DNA as much as it otherwise would. Now moving on to the dermis. The first layer of the dermis is the papillary dermis. And in terms of what kind of tissue is contained in this dermis, this layer, it's mostly areolar connective tissue. And areolar connective tissue is going to contain lots of different protein fibers, lots of different cells in it. It's going to have capillaries carrying oxygen and nutrients to the area. And also, we'll have some lymphatic vessels that will help with immune system function. So a lot of things are going on in the papillary dermis. Overall, it kind of has a binding and support function. Binding because it's connecting the epidermis with the other layers of the dermis. And protective functions because of the immune system cells that hang out here. For example, white blood cells, mast cells, and macrophages. The papillary dermis includes these undulations here. And interestingly, these bumps and grooves in the papillary dermis are what make up your fingerprints. So every person, even identical twins, are going to have unique fingerprints. Doesn't matter if all your DNA is the same, fingerprints are going to be always unique. Something I think is very interesting about that is the formation of the fingerprints, how the genetics influences the fingerprints a little bit, but there's also the environment that influences the fingerprint when the baby's in the womb the amniotic fluid, how it's flowing around, all that helps shape those fingerprints. And then in the last layer of the dermis, we have the reticular dermis. This is made of mostly dense, irregular connective tissue, and it contains lots of collagen fibers, has capillaries still plenty of blood and nutrients in this area, still lymphatic vessels. So in general, due to the dense, irregular connective tissue found in this area, it's really good at resisting tension in all directions. So, you could twist your skin, you can move it side to side, back and forth, all that. And the reason why it is good at not ripping apart could be partly due to this reticular dermis that contains this dense, irregular connective tissue. The collagen in this layer is really strong, and it's kind of like a spider web, like a three-dimensional spider web, and it's just able to resist those different forces. The reticular dermis also plays a protective role because there are some sweat glands that reach down to this area and basically involved in production of sweat, which contains chemicals that can help protect you from foreign invaders. And also you have oil glands that reach down to this area. And the oil glands also play a role in fighting off some foreign invaders on the surface of your body. There's some temperature regulation functions because, again, your sweat glands can go reach down to your reticular dermis, so that's where the production of the sweat's going to happen. And there's also some sensory functions because associated with the hair follicles, we have those hair follicle receptors in this area. So in the second section, we're going to look more closely at the different skin appendages. So we'll look at sweat glands, oil glands. We'll barely look at hair, and we're not going to discuss nails. So we're going to focus mostly on the first two skin appendages here. So sebaceous glands are oil glands. Oil glands or sebaceous glands have specialized locations. So we have different kinds of body maps here. So on the very left-hand side, we have a map of the sebaceous glands. And essentially what we have here is the kind of lighter blue color, or I guess that's blue. That lighter blue color is where you have a lower density of oil glands. So roughly from your chest down, or from the middle of your arm down and cutting off about at your wrist, you have a low density of oil glands, but they are there. The highest density are in these darker areas of the map. So from your head down to the bottom of your chest, going into your upper arm here, those areas have the most oil glands. In yellow then, where there's no blue, this yellow area, or these yellow areas, represent where there's no oil glands. The oils themselves empty out into hair follicles. We'll look at a picture of that in a little bit. And the oil glands are not working for your entire life. They actually will only start to begin functioning at puberty. And the function of oil is to not just lubricate skin and hair, but also to help kill bacteria. It has some antimicrobial properties. Then we can talk about sweat glands. So just a quick couple things about sweat. It's mostly made out of water, contains some salts, can contain vitamin C and some urea. also may have then fatty acids, but this depends on the age of the person, and also proteins. The sweat will only contain fatty acids and proteins after puberty takes place. So this is going to have some implications in how somebody becomes a little bit more smelly after puberty, so why people get worse body odor after a certain age. What ends up happening is these sweat glands, they start to release those fatty acids and proteins, and then the bacteria on the surface of your skin are going to metabolize those molecules. And guess what? It's not really you that's smelling bad, it's actually the bacteria that are releasing the scent from your body after metabolizing those macromolecules. So functions of sweat: dissipate excess heat through evaporative cooling, to excrete waste products, for example, urea. And also it helps reduce the pH on the surface of your skin, which will help inhibit bacterial growth. Some of the emissions, say like the fatty acids and proteins, can result in the odor production, which would be responsible for mating purposes, or could be. And there's more, but we'll kind of leave it at that. Now, two different types of sweat glands. So we'll have eccrine sweat glands and apocrine sweat glands. I'll say ESG for eccrine and ASG for apocrine. In terms of the maps, eccrine sweat glands is anything in green, which means you can find them anywhere on the body. The highest density are going to be in the darker spots, so the top of the head, the armpits, the palms of your hand, the bottoms of your feet. All those areas have the most eccrine sweat glands. And then the areas in light green have a lower density, but they're still there. In the next slide, we'll look at a picture of eccrine sweat glands, what they look like. Apocrine sweat glands, they're only going to be found in specific areas, so anywhere that's in this kind of brown color. So for example, your eyebrows, where your ears are, the nipples, the armpits, the pubic area, those are the 5 areas of your body where apocrine sweat glands would be. They empty out into hair follicles, so that's where the sweat will enter the hair follicle before it leaves to flow onto the surface of the skin. And these are the ones that begin to function at puberty. And so these are the ones that are responsible for releasing those fatty acids and proteins that get metabolized by bacteria. So putting these things all together, so here we have our hair follicle in the middle. We have our sebaceous oil gland kind of popping out of this hair follicle. So oil production occurs here. It will be secreted into this tube and then get released through the same hole that your hairs come out of your skin. So that's where oils will come out of. So once again, they'll help lubricate hair, but they'll also go on the surface of the skin to act as kind of an antimicrobial substance. The eccrine sweat glands are kind of these independent structures. So very convoluted tubes making the sweat, and then the sweat will go up the tube, eventually seeping out onto the surface of your skin. So these are the ones that are found everywhere in your body, and they're functioning since you were very little. Whereas the apocrine sweat glands on this side, those are the ones that flow into the hair follicle, similar to the oil gland, and they're going to release the types of sweat that are going to be containing fatty acids and proteins that result in body odor. So something I wanted to know about the hair is this response called the pilorector response. So if I were to spell it out, pilorector response, just like that. So the reason why your hairs stand up has to do with a smooth muscle that contracts, connecting your skin to the bottom of the hair follicle. This smooth muscle, its name is called erector pili. And so when it's relaxed, this erector pili, it's not pulling on the hair, so the hair can kind of relax and curve over more flat towards the skin. But when the erector pili muscle gets contracted, it pulls the hair follicle this way and also pulls that hair and helps it stand upright. This process of pilorection results in these goosebumps. Other people call them chicken skin, I think, depending on maybe what part of the states you've lived. There's different kinds of names all over for it. But those goosebumps are a result of the erector pili as well. So in the last section, just wanted to talk about a couple things having to do with UV radiation. So firstly, and not all of this is UV radiation, some of it is only kind of capable of fire, but let's go through the different severities of burns. So first degree burns are shown at the top right and described in the top left. This is when only the epidermis is damaged, and the result is some red and swollen skin. So most of us have experienced at some point in our life a first degree burn from just getting sunburned. But not everybody, right? Just kind of depends on your skin pigmentation in part. Second degree burns are going to be worse. So this is your epidermis got damaged and the upper dermis got damaged. If you got a second degree burn, a common symptom or common sign is going to be basically blisters on your skin, such as in that picture. Third degree burns are going to be the worst classification of burns, and this is going to destroy all the epidermis and all the dermis. And by destroying all the layers of the skin, essentially all the sensory receptors are destroyed, so you don't really feel pain in the areas that got burned. To fix this, somebody's going to need skin grafts. And this leads me to something important about burns. So burns are considered critical if, let's say if you have a second degree burn, if you have 25% of your body being second degree burns, so 25% of your body or more is covered in those blisters, that's considered critical. For a third degree burn, you only need to have 10% of your body to have a third degree burn to be considered critical. And the danger of getting a burn is that when you get burnt, you are exposing blood vessels to the atmosphere, and evaporation from these blood vessels can happen, and you essentially lose water really quickly, and the dehydration could lead to shock. Now, something tricky I want to point out here is, first degree, second degree, third degree, and third degree is the worst. But the system of ranking them is opposite of first, second, and third degree murder. So first degree murder is worse than third degree murder. So it's kind of a flipped severity scale. So just keep that in mind in case you're trying to make a comparison of how these are categorized. With UV radiation, over a long period of time, this can cause wrinkling. So what I have here in the picture is a truck driver who has been sitting in the driver's side of the truck, driving down the same road for years and years and years, right? Up and down the same road. And the sun always beating on the left side of their face while they were driving. And so you can see the aging of the skin on the left side is a lot more advanced than the aging on the right side. So what is going on here? Well, there's these specific protein fibers in the reticular dermis. Sorry, it's the papillary dermis. Apologies. So in the papillary dermis, the first layer of the dermis, you have some areolar connective tissue, which contains elastic fibers. And normally, elastic fibers help your skin bounce back. And if you were to pull on your skin, it kind of pops back into place. But over time, UV radiation makes those elastic fibers clump together, and they essentially start pulling your skin towards other parts of your skin, and thus you get wrinkles. Makes the skin look more leathery, and so this could be advanced by UV radiation. Now, something I want everyone to be learning at some point in my class is to be able to know how to detect skin cancer to a degree, or something that might be potentially a skin cancer. So just some really basic things about skin cancer, or just cancer in general. Cancer is an abnormal cell mass, and there are two different ways that you can classify tumors. One is benign. That's a tumor that doesn't spread. It's just encapsulated. And the more dangerous type of tumor is the malignant tumor. Malignant tumors are those that break apart and they can metastasize or move to other parts of the body. And this is dangerous because once those tumors can break off from each other and spread in your body, then you have more tumors in more parts of the body, and they can grow and push against organs that are really important for your body. Skin cancer is the most common type of cancer, so that's why I want you guys to be familiar with this ABCDE rule. This ABCDE rule helps you detect what might be a skin cancer or not. And I would like to preface this by, if you even think that you might have something that could be like a tumor, it's always good to just get it checked by a doctor. So this ABCDE rule is not like 100% guaranteed to tell you whether something's skin cancer or not. It's just kind of some guidelines. So always go to a doctor if you're curious about this. Don't try to self-diagnose yourself and say, oh, I'm not going to go to the doctor anymore because I learned that it doesn't fit the criteria for a dangerous tumor. All right, so anyway, that said, let's go through the ABCDE rule. So the A stands for asymmetry. And so what we're going to do here is compare a healthy mole on the left with a melanoma on the right to see what the difference in their symmetry is. So asymmetry is when, basically they have an irregular shape around them. So if you were to, say, put a dotted line down the middle, they're not symmetrical. The left side is not the same as the right side. If you did it with a normal mole, a healthy mole, it's pretty much a sphere, so left and right halves are the same. The B stands for border, so we have to think about the border here. Healthy moles, their border is going to be smooth and pretty even looking edge. But the border of a melanoma is usually more notched, a little bit more uneven. C stands for color. Healthy moles typically have just one type of color to them. Say it's a specific dark shade of brown, for example. A melanoma is more likely to have different shades of color. For example, here's a darker kind of brown area, and then on the right, there's a lighter brown. D stands for diameter. So healthy moles are going to be smaller, generally smaller than 5 or 6 millimeters. That's about the width of a number 2 pencil eraser, something like that. Whereas an unhealthy mole, which we would say this is a melanoma, that would be bigger than 6 millimeters in diameter. The E, which isn't shown in the pictures, stands for evolving. And this just means that if the mole appears to be changing over time, maybe it's changing in size, maybe it's changing in color, changing in symmetry. If it's changing, then, or as they say, evolving, which is kind of incorrect to say it's evolving, I guess, but anyway, if it's changing over time, then that is a hint that it might be melanoma and not healthy mole. So last couple points I want to make about this is that in order to be a melanoma, it doesn't have to fit every single one of these 5 criteria. It could just be that it maybe hits one of the criteria or 2 of the criteria, but the more criteria that the mole fits, the more likely it is that it's not a healthy mole. So for example, on an exam, if you're given a question like, this mole is, let's see, some mole is symmetrical, it has a smooth border, it has just one color, but it's really big, like 10 millimeters, and it's not changed too much over time, so it hasn't really evolved much. Now if I give you that description of a mole, and I give you a second description of a different mole that was asymmetrical, it had an irregular border, it had 2 different colors to it, it was a smaller diameter, maybe like 6 millimeters, and it was changing through time. Which of those 2 moles is more likely to be melanoma? And your answer should be the second one, because the second one met more of the criteria that match what a melanoma would be, even though the first one did match some of them, but it wasn't as much as the second one. All right, so we're going to stop there. Hope you enjoyed this quick video lecture. Thank you for watching.